One of the terms that we've mentioned previously in the class are proteins. And in this particular topic, we're going to explore much more detail about how the cell encodes information that determines the structure of these proteins. Proteins are made up of subunits called amino acids. And this figure is showing the general structure or composition of an amino acid. If you're not familiar with chemistry and chemical symbols, then let me review this for you. C is the symbol for the element carbon. N is the symbol for the element nitrogen. H is the symbol for hydrogen. O is the symbol for oxygen. These lines that you see joining carbon atom with oxygen atoms are called chemical bonds. These are forces of attraction that hold atoms together to produce molecules. Now, there isn't a element or an element with the symbol R. R stands for the variable group or the variable region. And this is the area of an amino acid that varies between the 20 naturally occurring amino acids. And in the table uh, presented here, you can see that we have the NH2 group, right? This is called the amino group. We have the C double bond O, single bond OH. That's called the carboxyl or the acid group. So you can see where the name amino acid comes from. It's combining a portion of the amino group name with a portion of the carboxyl or acid group name. Coming off of the central carbon, which has the hydrogen attached to it, you can see that chemically speaking, there can be a variety of different things. Each of the 20 amino acids has a different variable region or side group. You can see in this example, this is the side group for lysine. This is the side group for glutamine. This is the side group for aspartate. And again, each of the 20 amino acids has a different R group. The rest of the structure remains the same. Another aspect that's introduced in this topic is the concept of protein folding. And protein folding involves four levels, or each one associated with a particular term. So you'll hear the primary structure, the secondary structure, the tertiary structure, and in some cases, the protein is correctly described by having a quaternary structure. And this is also shown in another video clip that I've included, but I'd like to talk through it with you here because sometimes repetition is very helpful. So you can see that the primary protein structure is the sequence of amino acids. So each of these blue balls represents an amino acid. The amino acids are chemically joined together to produce the polypeptide or protein chain, and this is the first level of structure. The order of bases, nitrogen containing bases on one of the two strands of DNA, is responsible for specifying the order of amino acids. And that's going to be the focus of topics 4.2 and 4.3. What will happen is due to interactions between portions of the amino acids, the primary structure can twist and fold on itself to create one of two other types of structures. It's called a pleated sheet, or you'll sometimes hear the term a beta pleated sheet, or an alpha helix. Now, within a particular protein, you can have regions that will form a pleated sheet and other regions that will form the alpha helix. But what will happen is that interactions between the chemical R groups will cause the protein to twist and fold again. This is called the tertiary structure. And the tertiary structure you can see is due to side chain interactions. The R group represents the side chain. So within this, you can see there are regions that have the alpha helix, regions that have the pleated sheet, but we've added another dimension or another level of folding to it. The last level of protein structure does not apply to all proteins, but an example is mentioned in this particular topic that is applicable, and that is the protein called hemoglobin. Hemoglobin is a pigment found in red blood cells, and it functions to carry oxygen, which your cells need to produce energy. And this is an example of a protein that has a quaternary structure. What do we mean by that? It means that the functional protein is actually produced by more than one subunit of protein coming together. So in other words, you would have multiple tertiary protein structures that are interacting with each other to produce the quaternary structure. This is a figure representing the structure of hemoglobin. And hemoglobin contains two what are referred to as alpha chains. These are the red portions that you see here, as well as two beta chains, which are represented by the bluish purple portion here. Now, the green portions that you see are actually something else chemically. They're the heme group, because you hear the term hemoglobin. The heme group is the iron-containing portion of the red blood cell, or excuse me, of the pigment where oxygen will actually bind to. And so within a red blood cell, there will be millions of molecules of hemoglobin, each one of them capable of binding to 1, 2, 3, 4 molecules of oxygen. So to summarize, we looked first at a general amino acid structure. We talked about the amino group, the carboxyl group, the central carbon, and the side chain or the variable region. We said that it's variation in R groups that gives us the different amino acids, and the areas in blue are representing different R groups for different amino acids. And then we said when these amino acids are joined together, they'll produce different levels of protein structure: primary, secondary, tertiary, and in the case of some proteins, a quaternary structure. So I hope this video is helpful in understanding some basic aspects about amino acids and proteins.